So the last talk is from Simone Tiberi on BANDITS, a hierarchical model for differential splicing.
Okay, thank you. So let me briefly introduce myself. I actually am a statistician, so this might be a bit of a heavy talk for 5:30. So I feel like there are 9 more parallel sessions. And as a statistician, I gradually shifted towards biology, first in systems biology, and now I'm a postdoc in Zurich, and I'm working with Mark Robinson on statistical tools for bioinformatics. This one is a Bayesian tool for differential splicing on bulk RNA-seq. If you're interested, we also have a poster downstairs for a tool on single cell stuff to perform differential state analysis. So differential splicing, very intuitively, is the branch that studies how alternative splicing patterns vary in general terms between conditions. And there are actually a lot of methods to do that on RNA-seq data. But in our opinion, at least, they all neglect some of the key features of the data. So our idea was to fill the gap and present a method that would account for these elements. And I hope and think we've done it, and we've distributed it as a bioconductor package that's called BANDITS. So in the talk, I'm going to briefly introduce you to the differential splicing, mostly mathematically, and I'll describe you how we perform inference with our model and how we validate it in simulation and experimental data sets. So alternative splicing, you probably know it better than I do, is a biological process where a single gene can actually code for multiple proteins. And in RNA-seq, what you do is that you study how the expression patterns of the different mRNAs, called isoform transcripts, vary between conditions. So what you're interested in is the number of RNA-seq reads that originate from each isoform. But that's typically not observed directly. And so that's the biggest mathematical challenge when you look at transcript-level analysis. So if you look at this style example here on the top, where you have 2 transcripts, blue and red, and you have some reads. You can uniquely assign some reads to a transcript, like in this case, this blue one and these 2 are reds. But in most of the cases, you cannot uniquely assign a read to its transcript of origin. And so what most methods for differential splicing do is to use a 2-step approach. It's called plug-in approach in statistics, where you first use some quantification tools, typically Simone or Callisto, to estimate the number of RNA-seqs that come from each one of the transcripts. And so you might have some estimates like 4.4, 5.6, something like that. And then you then input these estimates in the tools. The problem with this approach statistically is that you neglect the uncertainty in these estimates. Well, you should ideally propagate that forward. Instead, what we do, and that's also an approach followed by a couple of other methods, is to look at what transcript each read is compatible with. That's typically called an equivalence class. And so again, in this toy example, you would have that 1 read is compatible with the blue transcript alone, 2 with the red one, and then 7 reads are compatible with blue and red transcripts. Just note that with our tool, BANDITS, you can actually align reads either directly to the transcriptome with Simone or Callisto, or also to the genome with STAR. So getting a little bit into the model. So take the canonical scenario where you have a gene that you're testing for differential splicing. You have k transcripts for this gene, and you observe n biological replicates, so n samples for a specific experimental condition. So that if you have ni counts for the ith sample at the gene level, what you assume is that these counts are distributed across your k transcripts according to a multinomial distribution. And this parameter here tells you the relative abundance of the transcripts. So 20% for the first transcript, 30% for the second, and so on. So what we do is that we use a hierarchical model. That's basically the Bayesian parallel of the frequentist model, of the mixed effect model in frequentist statistics, where basically each sample is associated to a distinct parameter vector. So you have a distinct PI for every individual. But then there is, of course, some, so this is to allow for basically over-dispersion between samples. But then there is, of course, some sharing of information between samples, and that happens with a common prior, which in our case is a Dirichlet. And this parameter here is called Hyperparameter, and it basically summarizes the information about the group, your experimental condition, and it tells you the relative abundance of your transcripts, basically average across your samples, and also via the precision parameter, it tells you how much this mean abundance changes between samples, so the degree of overdispersion. And since we use a Bayesian approach, we actually infer an informative prior for this precision parameter. And the information is basically taken from the other genes. But as I told you, I showed you that transcript level counts are assumed to be distributed like a multinomial, but we don't actually observe them directly. So we don't know how many reads originated from each transcript. So we treat this parameter as a latent state, where latent state in statistics are just unknown parameters that you're also going to model. So we are going basically to model the number of counts that originate from each transcript, and we use what's called a data augmentation approach, and basically we sample this parameter. So in practice, what we do is fairly simple. So we simply sample the allocation of these 7 reads to the blue and red transcripts. And a key thing about our approach is that basically, as I told you, there are several methods for differential splicing, but none of them at the moment uses a hierarchical approach that allows you for sample-specific proportions and at the same time a latent structure for the allocation of reads to the transcripts. And then there are also, in some cases, equivalence classes might also have transcripts from different genes because it happens that reads are compatible with transcripts that actually belong to distinct genes. So in this case, we just proceed in a similar manner where we not only sample the transcript allocation of reads, but also the gene allocation. And when I say that we do this sampling, we obviously do that iteratively in the MCMC. We don't just do it once. So modeling-wise, I'm not going to go into the formula details, but of course you want to study the full posterior, which is given by the latent states, the hierarchical and hyperparameters, and this is not there is no analytic solution for this formula. So what you do is that you use MCMC techniques to basically approximate this with a big enough sample. And since you cannot actually check the convergence of the MCMC manually for 10,000 genes or thousands of genes, we use an automatic stationarity test that checks that, ensures that your posterior chains have reached convergence. And so far, basically showed you how to, the description of the model and how to obtain the parameters for a given experimental condition. But now we want to test between experimental conditions. So say you have a canonical scenario with 2 groups, A and B. So the way we test for differential splicing is by looking at the mean relative abundance, so average across the samples on the group, for group A and B. So if there is no differential splicing, then these relative proportions of transcripts are going to be the same for all transcripts. If there is differential splicing under the alternative, there's going to be at least some differences, at least 2. The way we test for this system of hypotheses is basically to go back to the frequentist word, and we consider the difference between the mean relative abundance in the 2 groups, and we approximate this difference with a multivariate normal. So basically, we approximate the posterior with a multivariate normal, while the mean and variance covariance structure are just inferred from the posterior chains. And once you have this approximation, it's straightforward to actually use a multivariate wall test on this. And this allows you to test for differential splicing at the gene level. But we also provide a test at the transcript level, and we do it exactly in the same way, but obviously now you look at individual transcript, and so you have individual proportions, univariate normal approximation, and a univariate wall test. Now, something that we introduced that surprisingly was not, at least I'm not aware that it's out in literature, is an offset that basically accounts for the different lengths of transcripts. So basically, we distinguish the probability that reads map to a transcript, which is what's normally modeled, that of course depends on the length of the transcript, to the probability that a gene expresses a transcript. So if you take a very simple example where we have a gene with 2 transcripts, does this work? Okay, I'll use the mouse. So if you have a gene with 2 transcripts that have equal probability of being expressed, but obviously one is twice as long as the other one, then clearly you will observe, on average, 2/3 of the reads mapping the second transcript and 1/3 mapping the first one. So most methods for differential splicing work with and test this parameter here. So instead, we reparameterize, as in the formula here that I'm not describing, and obtain the probability of actually expressing the transcript. And we noticed that obviously testing this pi-t parameter, which is intuitive, I think, leads to slightly better performance. And also that's the parameter that we then provide to the user, so that they know what's the actual expression of transcripts and not the probability of reads mapping to it. So we then validate this in a few simulation studies. This is the most comprehensive one. It's a human simulation study where we do a 6 versus 6 group comparison. And we benchmarked it against like a bunch of methods. I mean, as you see, I told you there are pretty much several methods for differential splicing, and these are just like kind of the most recent ones. So the good news is that our method has slightly higher true positive rate for given FDR thresholds compared to the other methods. But also, interestingly, if you look at these circles, so basically these circles indicate you the actual FDR for the theoretical thresholds 1, 5, 10%. So when the user specifies that they want an FDR of 1, 5, and 10%, that's the actual FDR that you get. And our method BANDITS can roughly control for this, for the FDR. Well, as you see, all the other methods tend to deviate significantly from the canonical thresholds. And a similar story, if you look at the actual transcript level test, well, you know that there are fewer curves here because not all methods perform both gene and transcript level testing. Then we further looked at a couple of experimental data sets. The first one is a data set where, again, from a human genome, 3 versus 3 group comparison, where 82 genes were validated with PCR techniques, which is quite a lot. So things are a bit obviously rough here because we are analyzing 10,000 genes, but only 82 were validated. So we don't know the true state of the majority of genes. But you can still see that, again, like our methods got like slightly higher true positive rate. It's this one here. You don't really see it, but it's this one here on the top. And I think it's much more interesting to actually look at this first column here that tells you the median position of these genes in the ranking. So if you take the ranking of 10,000 genes, then you look at the median position of these 82 genes. And you can see that BANDITS pushes basically these significant genes more towards the top compared to the other methods. And finally, we also consider a data set where we perform a null comparison. So we're actually comparing 3 healthy patients to 3 healthy patients. And this is just to investigate false positive rates. And very briefly, here on the left, you have basically 3 dots, and they indicate you basically the number of false positives that you would get at the thresholds 1, 5, and 10%. And so ideally, you should have under the nulls, because this is a null data set, you should have 1 and 5, 10% of p-values below these thresholds. Our method is here, but you can see that our method, as well as some, the majority of methods, can control the false positives fairly well in this data set, with a couple of exceptions that have like a significant inflation of false positives. And you can draw exactly the same conclusions by looking on the right, where you have the false positives for a varying threshold. Similar story again if you look at transcript level testing. And I'll try to wrap it up in a minute. So we propose basically a Bayesian tool to do differential splicing on RNA-seq data. It requires a reference genome or transcriptome, according to whether you align to the genome or transcriptome, and it can actually input reads that you can align, as I said, in both ways. So you can either align to the transcriptome with Simone Tiberi or to the genome with star. It has a hierarchical structure so that you actually model the biological variability between samples, and it inputs the equivalence classes by using a latent variable structure and sampling the transcript allocation of reads and the gene allocation of reads when it's needed. We test for differential splicing at the gene level, but also the transcript level, so that you can actually also investigate what specific transcripts are significant in selected genes. And we introduce an offset to basically correct for the length of transcripts and basically provide the actual probability of expressing transcripts. And as we've seen in simulation and experimental data sets, it has very favorable performance. And lastly, from the computational perspective, I don't have plot for the actual benchmark because we're still working on it. But although we do full MCMC on thousands of genes, I think it is pretty competitive. Because, for instance, the full pipeline on a human, like a genome that I showed you before, so 6 versus 6 comparison, that runs like in less than 2 hours on a laptop, which, I mean, for a statistician, that's like a very good time. So, and in general comment, not just about my method, but I see that in the field, bioinformaticians tend to go for quick methods. But I think sometimes, you know, more mathematical methods require a little bit more time because they do more complex things, and I think it might be worth to wait like a little bit more. If you're interested, it's available on Bioconductor or as well on GitHub. And we don't have a preprint or paper, but we'll post it soon, I think less than a month. So if you're interested, it's going to be posted there. And of course, thank you for your attention, but also Mark, my supervisor, Charlotte, the full group, and other people who contributed with some helpful comments. Thank you.
Thank you for the talk. We have time for a couple of questions.
Thanks for the nice talk. I'm curious how does the differential splicing test goes with the base factor, for example, Savage density ratio test instead of a hypothesis test?
Yeah, it's a good question. I mean, it really is. So, where is the thing here? So when we basically get here and we want to test this system of hypotheses, we tried several things. Base factor was one of the things, but the problem is that base factor or posterior probability for model 0, 1 are not very stable because they were really heavily influenced by the highest value of the likelihood. So basically, you know, base factors were alternating between 0 and infinity, or the posterior probability of model 0 and 1 was alternating a lot between 0 and infinity. So it was not very stable. And furthermore, it requires to run the model twice under the null and the alternative, while we only need to run it once. So to be fair, we found this solution which was computationally efficient and it worked really well, so I didn't bother, you know, spending too much time on the alternative.
There's another question over there.
Thanks.
No, thank you.
Here, hello.
Hey.
Hey. So if I understand it correctly, you do like the expression estimate and differential expression of the differential splicing analysis like in one package, right? So when you are assigning the read allocation to exons that are shared between transcripts, like you were showing at the beginning of your presentation, are you considering also how is the distribution of reads on those exons that are not shared? Because I think this is very important information for distributing the reads on those that are shared exons. I haven't seen any indication that you are doing anything like that.
So I'm not sure I got your question right.
So can you maybe go to slide 10, I think it was, that one,
like,
right?
Okay, yeah.
So the F3, you have 7 reads, and
yes.
you are in a region in which you have a shared exon between the two.
But in these cases, for instance, you don't.
Yeah, but you can use the information on the exons that are not shared to infer how to distribute those reads. Are you using that information?
Yeah, I mean, absolutely. So the point is that I didn't show you like, well, you know, here I didn't really show you the actual, you know, probabilities in here. But yeah, definitely we use the full information, so all the reads. So in this case, it's trivial because you know the transcript they belong to. In this case, it's a little bit more challenging. But of course, we use all the 10 reads to infer the posterior probability of expressing the two transcripts. Yeah, I mean, absolutely. These are very informative because, I mean, the reads that are not shared, they're actually very informative because we know with probability 1 that they belong to a specific transcript.
So then it's not very different from other methods that are doing the same.
Like
RCIM, for example, is also doing that.
But no, I think, I mean, only CJBTIC, to be fair, does a latent variable allocation of reads that, I mean, only, no, it's let me try to find a way to say it clearly. No, it's different from the other methods in the sense that no method does this allocation and uses a hierarchical approach. And in particular, there is only one method that's called CJBTIC that does this allocation of reads. So no, it's distinct from the other methods in the literature, mathematically.
All right, thanks.
Okay. Thank you. Let's give our speakers of the session another round of applause. Thank you all for coming, and Francisco will close out HiTSeq.